Hi, this is Caroline Roper, and I'm going to give you a brief overview of our project entitled Using the Native Grapevine Immune System to Generate Pierce's Disease-Resistant Grapevines. So in this project, we are capitalizing on some data that we have from our collective work on Pierce's disease, where we've shown that treating grapevines with priming stimuli, such as lipopolysaccharide obtained from Xylella fastidiosa, potentiates a stronger defense response once Xylella is introduced into the plant, so in the post-challenge, pathogen-challenged prime state. And this is the elicitor that we are using. It is the lipopolysaccharide entity that I mentioned, and it's a major part of the outer membrane of Xylella fastidiosa and many other gram-negative bacteria. And because of this, it is a strong immune elicitor in plants and animals. Okay, so what we did is we extracted and purified LPS from Xylella itself, and first injected it in this experimental design into grapevines. So they either received LPS or water, and then were challenged with Xylella fastidiosa, or were challenged with just plain buffer as the controls. Okay, and so these were considered the primed and pathogen challenge, and these were the primed and mock challenge. And these were what we call naive because they did not receive any immune elicitor. And once we allowed those plants to propagate in the greenhouse and develop Pierce's disease, we saw that the primed and challenged plants were healthier than the naive and challenged plants, and the negative controls did not develop Pierce's disease at all, as was expected. And so you can see this either in an area under the disease progress curve, where we see that there's less disease in the primed plants. There's also less Xylella fastidiosa pathogen titer in those plants. And internal symptoms, we see that there is significantly fewer tylose obstructions in the xylem, which is a hallmark of Pierce's disease development internally. So not only did they have less external symptoms, they also had less internal symptoms. So we wanted to understand what was the mechanism underlying this, and is it something that we can capitalize on to develop Pierce's disease resistant vines. And so we did a global transcriptomic study where we set up the plants in that same experimental design I just described, but instead of letting them go through the disease cycle, we harvested their petioles at different time points during that disease cycle and looked at what genes were being expressed at those different time points under naive or primed situations. And what we saw was that there were differentially expressed genes in the plants that received the priming stimulus versus the ones that didn't. And some of those were shared between local, so near the point of inoculation, or even 10 nodes above the point of inoculation at a systemic location. And these were at very early time points post pathogen challenge, so either 4 hours, 24 hours, or 48 hours. And so there's a significant differential response in the primed plants versus the naive at both local, but also what was astonishing to us is that all the way up at the top of the plant, even as early as 4 hours, the plants were sensing and responding to Xylella in a different way than when they were primed with LPS versus when they were not. And so we took a dive into these genes, trying to figure out, is there anything that we can utilize to generate Pierce's disease resistant vines? We did do a weighted gene co-expression analysis to show which genes of the differentially expressed genes in the primed vines were shared between the local and systemic tissues and also were different. And so the ones that we chose to focus on in this project were ones that were shared between the local and systemic and were differentially expressed in the primed plants. This is just a list of some genes that you can again find in this manuscript. These are the ones we're focusing on in this project in terms of expressing them transgenically and also trying to edit them out using CRISPR-Cas9. So that's the end of my presentation, and I thank you for your time.